To those affected by mitochondrial disorders, UMDF is here for you. Since 1996, we've worked to provide support, fund research, and create awareness.
Our mission is to promote research for cures and treatments of these diseases and to give support to affected families.
We want future generations to grow up in a world without mitochondrial disease.
I'm playing for the United Mitochondrial Disease Foundation.
Look at that. Nice check for you.
Over the years, our community has grown and our message has sparked a movement.
Hey everybody, it's Maya Mialik. Have you ever heard of mitochondrial disease?
Together, we've been able to make a difference for patient families around the world. Today, we are helping thousands of people navigate their mitochondrial disease journey. We're here, listening and providing support. We're here, educating and connecting patients and healthcare providers. We're here, finding and funding the best cutting-edge research. We will not rest until we find a cure. Join the movement at umdf.org.
Welcome, everybody, and thank you for joining us for the clinical trial update with Cyclerion Therapeutics. Today's speakers will include Chad Glasser, the Director and Clinical Scientist and Clinical Research at Cyclerion, as well as Dr. Marnie Falk with Children's Hospital of Philadelphia. Our moderator today will also be Dr. Phil Yeske. Just a few housekeeping items to get started. I want everybody to be aware that this session is being recorded. Throughout some of the UMDF slides that you'll see here is a QR code. For more information, please just use your mobile phone, take a picture, and it'll open up to the appropriate site on umdf.org. This one particular has Zoom help if you need any help with Zoom functions. So under your view options at the top of your screen, we do encourage you to select side-by-side mode so that your presentation fits your screen. All of our participants today are muted. If you have questions throughout this presentation, please be sure to use the Q&A box. We will try to answer those at the end of today's session. If you happen to have any technical issues throughout the session today, please just simply raise your hand and one of our staff partners will respond through the chat feature. Only UMDF staff will be using that chat feature, so be on the lookout if you do happen to raise your hand. Additionally, closed caption is available. You can toggle that off and on at the bottom of your screen in the menu section. And today we're going to be having polls. We ask you to please participate in those polls. Your answers will remain anonymous, and those calling in on phones will not be able to participate. Just a few UMDF updates before we get started. We encourage you to check out the latest and greatest updates relating to the coronavirus, specifically about the distribution of vaccines. Pardon me. And of course, be sure to check out Mito University, where a lot of our video content, including the recorded version of today's webinar, will be available for future use, as well as some of our content, a lot of our content from this year's past symposium. We also, many of you know Margaret Moore, our UMDF Support and Education Associate. If you need any help whatsoever or need to connect with her, the 800 number is here. We also can connect you to peer-to-peer support. We have almost 100 UMDF ambassadors in 29 states, and we also have programs for our teens and young adults. And our Fall Energy for Life walkathons will be kicking off here in September. We encourage you to walk your way with one of our scheduled events or in our national Walk Your Way event. And we want to thank Cyclerion for sponsoring our New England virtual walk and joining a team and forming a team, I should say, with Team Cyclerion. And our Ask the MitoDoc webcast series will be starting up again in September, so be sure to check the calendar of events and join us. And last but not least, we are going to have a poll session right before we, poll question, excuse me, right before we get started here. Before I turn it over to Phil Yeske, I am going to stop sharing my screen and I am going to launch a poll question. And it's basically a demographic poll. And we just ask that you, hopefully everybody can see that. Relaunch that. Answer this as best as you can. So what best describes you? Are you a caregiver, clinician, family member, industry partner, patient research, or other? Give just a few more minutes.
So Beth, maybe while we wait for those poll results to come in, I appreciate your mentioning the COVID webpage of UMDF. I just wanted to let the patient community know that I'm working actively with our Scientific and Medical Advisory Board to post an update surrounding back to school guidance. We know that this is a really hot topic right now within the community for very good reason. So we thought it was time to bring the SMAB back together, think about what as healthcare professionals would recommend for our patient community and how to approach that. So we're finalizing that. We hope to have that posted early next week.
Great, thanks, Phil. All right, I'm going to end the poll and share the results with everybody. And we can see that majority of folks are industry partners, followed closely second by a family member, and researcher tied for second place, clinician and caregiver.
I want to know who those others are.
Oh yeah, I did. Thanks, Phil. That's the majority. Others, yes, interesting. Thank you. All right, I'm going to stop sharing. And I am going to turn that over to you now, Phil.
All right, thanks, Beth. So, as Beth mentioned, today we're here to discuss a clinical trial active in the mitochondrial disease community. The sponsor of the trial is Cyclerion. We're going to be joined by Chad Glasser, who is a director of clinical research at Cyclerion. We're also going to be joined by Dr. Marnie Falk. I think most of you know her in her role at Children's Hospital of Philadelphia in the Department of Pediatrics, where she's now distinguished chair. She's also a key investigator on this trial, and we thought worthwhile to really bring Marnie to the table to share her thoughts as an investigator in the trial, to share with the patient community about being involved in it. So welcome to you both. Great to see you, Marnie. Great to see you, Chad. Chad, I'm going to turn it over to you to begin with a brief presentation about the company and the trial itself, and then we'll turn to Marnie for her comments.
Great. Thanks, Phil.
Sure.
Beth, can you project my slides, please? Fantastic. So it goes without saying we're super excited to have this opportunity to talk to the external community, especially patients, and share what we're doing in this space. So again, I'm Chad Glasser. I'm in our clinical research group at Cyclerion Therapeutics, and again we have Dr. Marnie Falk with us, who, as Phil said, doesn't really need much of an introduction, but she's played a key role in our study basically from the very beginning. Has been involved since we were talking about early study designs, and we're also doing some exciting work with her in the preclinical setting as well. So towards the end of the presentation, I'll turn it over to Marnie to provide her thoughts on our study and some thoughts on 6463 and the patient population as well. So for this portion of the talk, the focus is really on this trial that we're conducting, and it's an ongoing study where we're evaluating the safety of 6463. Sometimes it's referred to as CY6463, depending on the study, but we're really interested in its ability to treat neurological symptoms in patients with MELAS. So the first slide, please. So I think the first slide is just an overview of our company, Cyclerion. So we're a very small CNS-focused company, CNS meaning central nervous system. We're based in Cambridge, Massachusetts, and our expertise is really in the pathway, the nitric oxide pathway, which I'll take you through on the following slide. The first key point from the slide that I want you to take away is that this is not a new pathway. This is a very well-researched, very well understood pathway that's been modulated successfully outside of the CNS and has been validated as a druggable target. In fact, there's even two therapies that modulate this pathway the same way that 6463 does, and they are approved and widely used today. Actually, could you go back to the previous slide, please? And then the second point is that 6463, which is an oral once daily therapy, is the first molecule in development that's able to stimulate this fundamental signaling pathway, not in the periphery or in the blood or the vasculature, but in the brain and in the CNS. And this pathway has been linked to a range of mitochondrial disease pathologies, and that gives us confidence that this drug could potentially have a place in the treatment paradigm. That's also supported by other drugs that many of you are likely familiar with, such as Arginine and Citrulline, that also kind of work through this pathway and support our belief that this could provide additional benefit. And then the final point is that what are we actually targeting? What aspect of the disease are we targeting with this therapy? And you can see that the phrase CNS symptoms is mentioned a couple times on the slide. And what we're referring to when we say that are things like trouble concentrating, trouble remembering things, or trouble focusing, anywhere from that to issues such as seizures, stroke-like episodes, and headaches or migraines. And so our focus in this area is really justified by the preclinical data that we've generated, what we even know about the pathway clinically, and also even some of the data that we've generated in the clinic, which I'll share with you a little bit later. So this is a little bit of a different approach than I think many of the other therapies that have been evaluated in this setting, or current and past therapies, many that have targeted things like general fatigue and muscle symptoms. So we feel like we're different in that sense. And our goal with this initial study is to confirm safety and tolerability and generate some early biomarker data that would show that we could potentially have some impact on these early CNS manifestations of disease. So now next slide, please. So pathway slides, I know there's a lot on this slide, and I'm not going to go through every bit of it, but the relevance of this pathway, I just want to say, in brain physiology is supported by a vast amount of research, preclinical work and academic work. I know that, again, this has a lot of information on it, but there's three key points, I think, from the mechanism that I want to bring to your attention. So how does the pathway start? Where does 6463 bind? And then what does that binding do, and how does that molecule impact a range of downstream processes? So you'll see the start of this pathway is a molecule that many of you are likely already familiar with, as I said, especially if you have MELAS or know enough about the MELAS diagnosis, and that's arginine. So arginine is often used as a nitric oxide precursor because it's converted to nitric oxide by an enzyme called nitric oxide synthase. And then when that nitric oxide binds to a molecule, which is the red molecule called SGC, that binding basically increases the production of a molecule called cyclic GMP or cGMP. And that's arguably the most important molecule in this pathway, because what it does is it regulates a range of downstream beneficial effects, and those are all listed here. This is anything from neuronal health and the neuron's ability to communicate with each other, to anti-inflammatory effects, to bioenergetics and energy production, and even on vascular function. So what happens when we add 6463 to the mix is that it binds in combination with nitric oxide to that red central molecule SGC, and that binding stimulates that red molecule's activity and increases the concentration of cyclic GMP. And as I said, that's really the molecule that we're trying to increase the concentration of, because it has those positive beneficial downstream effects. Next slide, please. And I apologize, this slide has animations in it, Beth, so I'll tell you when to advance each one. So this is where we are in development with 6463. And I think the key thing I want everyone to take away from this slide once we're done with it is that we actually know quite a lot about this molecule, even though this is our first patient study. We've conducted a range of preclinical animal disease models, as well as a couple phase 1 clinical studies in humans. So if your worry is that you might be a guinea pig and that we know very little about this molecule, that's we don't believe that that's the case. But like any new molecule, we start in the preclinical setting. We test the drug in a number of pharmacology and disease disease models. And the goal with that is really just to see if we have a drug. And specifically for this molecule, are we achieving concentrations in the CNS? Are we modulating this pathway in the brain and the CNS at levels that we think make this drug potentially viable as a drug to test in a clinical study? So we did that successfully. If you could advance the slide. And the next phase was our first clinical study. This is our first human study, which we completed in January of 2020. We enrolled quite a few patients, 110 healthy volunteers, and we concluded that 6463 was safe, was well tolerated across a really wide range of single doses and multiple doses that were given over a couple weeks, and that the targeted concentrations within the CNS were measured to be similar to the concentrations that were associated with efficacy in animals. So we felt that this satisfied really the key objectives to move forward and further in development. So next advancement there. So our next study, we took our optimal dose from our first human study and tested it in a second phase 1 study, which we've been calling our translational pharmacology study. And this was conducted in elderly individuals, so folks that are 65 years of age or older. And in this study, we were able to confirm the safety and tolerability profile and some of the drug properties that we observed in the previous study. But the aim was to take a little bit farther and to look at whether we are able to demonstrate any sort of CNS activity that we've been able to observe preclinically. And what we saw were effects across a range of biomarkers linked to a number of neurodegenerative diseases, including mitochondrial disease and MELAS, and I'll talk about that in a bit. Next part of the slide. So where are we now? Obviously, we're kicking off patient studies, and we're really excited to bring this molecule into populations where it could actually provide benefit. And the goals of these studies are to continue to demonstrate an acceptable safety profile, but also to start to measure some early impacts on disease using some objective biomarkers. Next slide, please. So as I said, supportive of why we're excited about this molecule in these patients with mitochondrial disease are the results from the study that was most recently completed, our translational pharmacology study, which again was designed to look at CNS activity of 6463. And so the key data is on this slide. There's a lot of data from this trial, but we've tried to focus it a bit. But we used an imaging technology called EEG, or electroencephalography, which is it's a cap that you put on a subject's head, and it measures changes in brain electrical activity. And what we were able to conclude is that we are improving brain neurophysiology, which is evidenced by the figure on the slide. And this is showing an increase in an EEG signal called alpha power. And you can see it's in the back of the brain. So just to orient you, this is a top-down view of a person's head, and so the nose is on the top. You can see that the area of effect is really focused in the back of the brain. In terms of what this means clinically, alpha power is a brain frequency that is widely accepted and associated with attention and higher level processing. And we know that it's also diminished with aging and in many neurodegenerative diseases where cognition is impacted. And then two additional findings we think were exciting and showed promise were reductions that we observed in some neuroinflammatory biomarkers and improvements that we observed in eye movements, which are often evaluated early in these types of clinical studies as a way to measure motor function and measures of attention and arousal. So altogether, all of these speak towards this drug's potential to impact cognition and impact brain neurophysiology. And I think the most exciting part is that this occurred in a healthy elderly population and over the course of 15 days. So for us to move into a patient population such as MELAS, I think it really excites us, and we're hoping to see some additional effects on the brain in this study as well. Next slide, please. So specifically, why do we think this pathway works in mitochondrial disease, and more specifically MELAS? And I've kind of been alluding to this as I've gotten to this point in the presentation, but there's clear links between reduced signaling of this pathway and a number of mitochondrial disease manifestations. So things like cognitive impairment, disrupted blood flow in the brain, oxidative stress, inflammation, and especially stroke-like episodes, which of course are key aspects of the MELAS MILOS diagnosis. But we know that mitochondrial disease impacts a wide range of tissues and organ systems, but we've been hearing that CNS symptoms are often associated with some of the most severe and really most debilitating aspects of the disease, and they affect approximately 80% of individuals with mito. So even outside of MILOS, this is still an issue of high unmet need. As I said previously, this CNS manifestations of disease and neurological symptoms is a fairly vast bucket of things. It can be anywhere from learning disabilities and clinical dementia to strokes, headaches, and seizures even. In terms of the precedent that I spoke to earlier with Arginine, this increased our confidence that this could be a drug that could work in this population. So intravenous Arginine is used in the acute treatment of stroke-like episodes and has been shown to improve outcomes in MILOS patients. I think where the jury is still out is whether if you give Arginine in a chronic setting, orally, daily, or even citrulline, which is converted into Arginine, we're not too sure if it really prevents stroke-like episodes. It kind of depends on who you talk to. But I think the general conclusion is that there's still room for benefit, that there is potentially some anecdotal benefit there, but there's ways that we could improve, tolerability. Arginine isn't necessarily tolerated super well, requires much more frequent dosing. So we believe that 6463 could really improve upon kind of this early precedent that we've observed with some of these nitric oxide precursors. Next slide, please. So one of the questions we get a lot, because we were constantly compared to Arginine because it impacts this pathway, it's part of the pathway, is how are we different? So I wanted to spend a little bit of time on that. So the pathway is depicted here again on the right. So at the start, again, is Arginine, which is converted to nitric oxide. And as I said, citrulline is often used as well because it's converted to Arginine and has some other advantages in terms of dosing. But when you give these nitric oxide precursors, what we're in essence doing is we're quote unquote feeding this pathway at the beginning, and we're increasing cyclic GMP, but we're also kind of limited by the pathway's inherent ability to create cyclic GMP. In contrast, what 6463 is doing is it's stimulating the pathway. It's binding in combination with nitric oxide to that central molecule SGC, which is again the red molecule, and it's amplifying nitric oxide signaling. And what we've shown preclinically is by stimulating SGC, we're able to increase the concentrations of that really important molecule, cyclic GMP, well beyond what you're able to achieve if you were to just give Arginine or citrulline alone. So the key takeaways in the box in the blue, because of how 6463 binds with nitric oxide to SGC, it actually has the potential to work in combination and synergistically with nitric oxide precursors like Arginine and citrulline. So for patients that are out there that are interested in the study and are really comfortable with their current dosing regimen and their Arginine and citrulline regimens, we do allow you to continue on those regimens provided that those are stable and you've been on them for a certain period of time because of that potential for our drug to work in combination. Next slide, please. All right, so to move on to what most people are probably interested in, and it's more details about the study that we're conducting in patients with MILOS, which I will say throughout the presentation is actively looking for participants, and that's one of the reasons that we wanted to have this webinar, is to increase awareness of the study. So this is our first study in MILOS, but as I mentioned, we do know a lot about 6463 from the clinical studies that we've already completed to date. So the goal of the study is to confirm and further evaluate the favorable safety and tolerability profile that we've observed to date, but it's also to learn about what 6463 is doing and what effects could it potentially be having in this shorter study on mitochondrial disease and mitochondrial function. So to do this, we've included a range of exploratory endpoints and assessments, and I'll talk about those in a bit. But as a participant in a trial like this, I'm sure many of your questions are about what you can expect for the trial and how will you go through, what's the visit schedule. So we do want to talk a little bit more about that. So one of the good things about the design, if you are a participant, is that this is what's considered an open label study, meaning that if you enroll, you will automatically get 6463, and you'll get it orally, you'll get it daily. There's no sugar pill and there's no placebo. In terms of how long you'll be involved in the study, the treatment period itself, or the amount of time that you'll be receiving the drug, is 29 days. So it's relatively short. It's a relatively short time commitment compared to some of the other studies. And in terms of the visit schedule, there is, of course, an initial on-site screening visit at a clinical site. The site will evaluate you, determine if you're able to join the study, if you're eligible. And then you would come back after a period of time for your first dose of 6463 in the clinic on what we call day 1. And then you'll be sent home with the drug. You'll take it daily, orally at home. And then at the very least, you'll come in again at the end of treatment on the last visit on day 29. There are additional check-ins at week 1 and week 2, which you do have the option to either complete at home if a nurse is available and can visit you if that's your preference. That's a conversation you can have with the site and with your investigator and the study staff. But there's the option to have that study be completed at home as opposed to having to go into the clinic for that. There's also a final follow-up visit, which is a couple weeks after that last dose on day 29. And we're actually working on a way to make that visit also kind of hybrid and can be partially completed or completely completed at home, if that makes sense for the patient, again, in conversation and collaboration with the clinical site. The length of the visits of the study is a question that we get a lot as well. So the range can be anywhere from 4 hours for some of the shorter visits to 8 hours for the longer visits. One of the longest visits is day 29, of course, because this is really the last day of getting the study drug, and because this is when most of the really key assessments are going to be performed to see if 6463 is having an impact on your disease. In terms of the size of the study, we're enrolling up to 20 adults. Adults meaning 18 years of age and older currently. There needs to be confirmation of a genetic mutation associated with mitochondrial disease. There also needs to be evidence of clinical disease and a clinical phenotype, so neurological features that are consistent with MELAS. And then, as I mentioned, one of the reasons that we are hosting this is that we are actively looking for patients, and we do have a number of clinical sites that are active and doing outreach and trying to find patients and increase the size of the funnel. These are all very key, educated, important sites in the mitochondrial disease community. We have them on a map on the following slide, but they're also listed here. And then as a company, we're very mindful of how challenging some of these studies can be for rare disease, for severe populations. So we're trying to make the study as easy as possible for patients to participate. Some of the things we're doing is we have concierge services that we've included as part of the trial. This means that they will help with patients and families scheduling travel and transportation to and from the visits. As I said, we do allow for at-home visits for certain interim study visits, so you don't have to travel to the study center for every single assessment. And then if you do live a certain distance away from the sites, we are also offering reimbursement for travel as well. So flight reimbursement, hotel stays if you need to stay closer to the site for the duration of the treatment period. Again, we know that these sites ask a lot from patients and from families, so we really want to do everything we can to make it as feasible for the largest number of participants to get involved. And next slide, please. So in terms of the assessments and what we're actually going to be doing to participants that are enrolled, as I said, safety and tolerability is still important. That's really the primary endpoint. We want to confirm what we've observed to date. To do that, we're going to do standard, safety labs and adverse event reporting that you do in these typical kinds of studies. But we also have a range of exploratory endpoints that we've added to the trial to look at early impacts on disease. And these were really chosen based on the preclinical data that we've generated and also supported by some of the clinical data that we've observed to date and the domains that are relevant to mitochondrial disease. So of course, want to test 6463's ability to impact mitochondrial function. So we're evaluating a range of biomarkers that are related to mitochondria and energy production. Some of these biomarkers can be measured in your brain using MRI. Some can also be measured in your blood using blood samples that'll be be drawn. The MRI procedure is non-invasive. There's no contrast. It's not very complicated, but you will be asked to relax and stay still while in the machine for the total scan time, which is between 45 minutes and an hour. We'll also be using these images of your brain to measure how it's being perfused with blood, and also the blood samples to look at biomarkers that are associated with neurodegeneration. And then finally, I've also mentioned cognition throughout the talk. That's a high interest to us based on what we know about our pathway and what our company is focused on. So we've included a couple of cognitive questionnaires for patients to fill out personally and give feedback on their experience. So these are things that are looking at cognitive fatigue and cognitive function. And those are interspersed throughout the study visits. And then finally, I do want to briefly mention another exploratory endpoint in this trial, which is measuring drug concentrations of 6463. Actually, you can go back to the previous slide for one last point, if you don't mind. The molecule, does get into the blood, but more importantly, because we know we're targeting the CNS, it's getting into the central nervous system and into the brain. And one of the goals is to also measure concentrations of this drug in the brain and in the central nervous system. So because the cerebral spinal fluid is a fluid that surrounds your brain and your spinal cord, in order to get that sample, procedure called a lumbar puncture needs to be performed. And this is basically where a needle is inserted into the lower back between two vertebrae to extract that sample. To date for this study, that has been a requirement per protocol, but we're actually in the process now of allowing participants to opt out of a lumbar puncture if that is really the sole reason that they are not willing to consent to the study. We do hope that most participants will agree to that procedure because it will tell us a lot about whether we're achieving those targeted concentrations, and it will also support some of the other endpoints if we see changes in those. But we do appreciate that this is one of the more invasive procedures to include in a protocol, and we really don't want this to be the sole reason that we're not able to continue enrollment in the trial. So we want to make it a little bit more accessible. So now my final slide, please. So, this I hope the presentation kind of takes you through who we are, what our focus is, what our molecule is intended to do, and also, really why neurological symptoms of mitochondrial disease is of increased interest to us. So before I turn it over, the mic over to Dr. Falk to provide some additional comments, I think we have a poll question, which we'd like everyone to answer. So I'll let maybe Beth pull that up. But so for these questions, I think we have one question now and then a couple a little bit later, but these are really meant for patients to answer. I'm not sure how many patients we actually have on the call based on the results from the initial poll. But if you're not a patient, we do ask that you answer kind of on behalf of the patient that you may care for, or the patients that you're closest to, or that you represent, or you're most aware of. So the question, sorry, I was waiting for it to pop up. So in terms of quality of life, so to what extent do neurological symptoms of disease, and as we've been saying, this ranges from anywhere from seizures, stroke-like episodes, cognitive impairment, headaches, etc. How much do those impact your quality of life? So to a very large extent, to a large extent, neutral, no comment basically, little extent, or very little extent. All right, so we're happy to see this result from those that did participate. It looks like the most common responses to a very large extent, and the rest were a large extent. So this is great. This validates, again, our focus, and hopefully we'll be able to further inform ourselves whether 6463 could have an impact there. All right, so without further ado, I'll turn it over to Marnie. Marnie, would you mind commenting maybe on your perspective on some of these neurological symptoms that you've seen in your patients and our molecule and our study, as well as maybe some comments on some of the preclinical work that's exciting that we're doing with you?
Absolutely. Well, thank you, Chad, and the Cyclerion team, and of course the UMDF for hosting this and for having me today. I think it's great that we're able to have this conversation, directly with the families and share with them the rationale. You know, as a scientist, we see a lot of things behind the scenes and we can't always share, but I do think it's critically important that, you know, we share why this is so exciting. So it's also really impressive how fast this has moved. So it may not even have been on some people's radar. I think the history is really neat. Cyclerion became aware of mitochondrial disease and actually came to some of the meetings we were having on therapeutic development when we started brainstorming together about, you know, this would be great to have an FDA approved medicine to improve the cognitive function in mitochondrial disease. That's sort of the holy grail. A lot of the other focus is important. It's focusing on exercise and energy and other symptoms, but there's really not a lot going on to actually help the neurologic problems, which are significant. I think the wording sometimes is confusing for this particular study. You know, it says MELAS, and I think our field has been challenged and you know, tasked with understanding all the different names of all the different subtypes of disease. So I think a lot of people hear this trial and think it's only for people with the 3243 A to G mutation in the mitochondrial DNA. And while that is a common cause of MELAS, and certainly people with those symptoms, with that mutation can have neurologic problems, some don't. So you wouldn't get in unless there were neurologic problems. And then there's many other types of mitochondrial disease, both in the mitochondrial DNA and in the nuclear genes, that cause neurologic problems. So it's really more about targeting the neurologic problems. And it's not even just the brain, it's also the peripheral nerve. So I think some of the opportunity here is to really understand that this is really directed to treat the hardest part of mitochondrial disease. And I'm always humbled to remember that it wasn't so long ago, you know, 7-8 years ago, that the UMDF's mission statement changed to include therapies as a goal, because mitochondrial diseases were thought to be so hard to treat. So to have a company really move forward with a brain-focused therapy is really important. And I definitely wanted to support it. What I would say after we met, what was really exciting is that they didn't just design a protocol and drop it in the mitochondrial disease community's lap. They actually reached out to us and the experts who know the disease to review their data and to start thinking about it really carefully and conscientiously in the context of mito disease. So they looped in, you know, some really great doctors, you know, at our center, including Dr. Goldstein, whom many of you know is a world-class pediatric neurologist. They brought in the neuroradiologists themselves who really understand the brain function and testing probably better than anybody. And really, we worked for about 2 months with a pretty big team to design the study. So the study was designed by mitochondrial disease clinicians and experts. And the other thing is that they really took care to listen. You know, the LP, the spinal tap, the lumbar puncture, that's being done under like the utmost care. It's being done under fluoroscopy, you know, by people who, you know, are using ultrasound guidance. So they really listened to some of the concerns and some of the challenges, and really, I think, designed a very thoughtful study. And then in pretty fast time, I want to say in 8 or 9 months, Chad, this study was live. So I think the focus and commitment has been really a model to follow, you know, the desire to take a therapy, understand it really carefully, get some data, and then do it really safely, but to do it with the mitochondrial community. And so I think this is a big opportunity. You know, this is a phase really 2 study, very early in the mito disease patients. Everybody gets the drug, like Chad said, which is kind of neat, but it's still an early study. And really, if this drug is going to be FDA approved, there will need to be another study. So, and it will be probably much bigger and it will be placebo controlled. So this is actually a pivotal study to decide if this drug is going to be developed for the mito disease community, right? So that's why it's so important that we all work together. I know there's 5 sites already. I think I saw a comment, you know, how do we get people in from other parts of the country? I think that is a pickle. I'll leave that one to Chad, except to say they're very aware that people with mitochondrial disease have a lot going on. And when you have neurologic problems, including mood problems, you know, depression, anxiety, sometimes cognitive clouding, really processing, it's hard to like organize all of life and be in a trial. So I think the ability to really connect with families and hear what each one needs is really important. So this concierge service, you know, I think means different things to different people. And I would just encourage people to reach out to the coordinators and talk about what their actual needs are. You know, is it travel for the month? Is it travel a couple of times? Is it babysitter? Is it a car service? You know, what is it that you need? And I think they really understand that this population has a lot going on and needs a lot of support. So I think that's also big, not just like figure it out, and if you show up on our doorstep, we'll give you the medicine. It's really very, very collaborative and supportive. So I think all that's great. And then on the laboratory side, this is not a nutraceutical. This is a medicine being developed as a drug. And that's exciting because it's also more potent. So the potency is, how much you have to take to have an effect. And they know that it gets into the brain. So a lot of medications, that's not known. And if it is, it's usually it doesn't get in. So it really is a molecule that's been developed as a drug for people. And that's exciting. And, just like we know aging is partly a mitochondrial disease, to see that they had an early trial where they actually, in only 24 people, saw significant improvement in cognition with age, that to me is very compelling, that there's something going on cognitively. We all know that cognition has a risk of falling with age, but to actually have something that prevents it or helps it, that's kind of the holy grail. So again, I know mito disease and aging aren't 100% the same, but there's an awful lot of similarity. And I think there's enough here that we need to be pushing forward together. So our site, of course, helped design it, is really very eager to support families. It is a fairly short trial if it's your first time getting involved in clinical trials. So a lot of trials are 6 months, a year, sometimes even longer, 2 years. So this is something that potentially in about 4 to 6 weeks, however, by the time you're screened and come in, and you can stay on your medicines, which is another key thing we know the families want. You don't have to come off of Arginine, you don't have to come off of your mitomeds. So that's pretty neat. There's of course inclusion exclusion criteria you have to be screened for, but that said, it's a pretty quick trial, and after this trial, after a certain number of days, you can go into another trial. So I think that's important, right? That our community and the UMDF has really encouraged that this is a village, right? It takes all of us to work together to design the trials, create the trials. There's now one open at 5 leading sites. There's ways to get in. You don't have to have classic MELAS. You can have migraines and definite mito disease, or other aspects, seizures and strokes and things that are really hard to treat. So I am just really hopeful that we'll learn if this is going to be a therapy. And if so, then this is a pretty short trial for us to figure it out. But I think the company's been very responsive to families when they brought up concerns about getting an LP. Not everybody wants it, or about the timing. I think it's also important. This trial is not going to be open forever, right? It's a pretty small trial. So the quicker we can get people in and through the trial, the quicker we can get to the actual phase 3 trial, which would lead, assuming that there's safety and benefit in this first trial, which would lead to the drug becoming widely available if it was positive. So I think I'll leave it there. I think that's most of what I wanted to say. But certainly there's such great coordinators involved here, and people have put their heart and soul into doing this. So we're trying to bring forward a therapy that seems potent, that seems to get into the brain, that seems to make thinking better. We're hopeful that it'll have an effect on the rest of the problems, but this is how we're going to learn. So I agreed, of course, to participate because I am excited, to see drugs come through for the hardest problems.
Yeah, so thank you, Chad. Thank you, Marnie. Great information. I think a lot to unpack. Maybe through a brief Q&A period, we can hit upon some of the questions that have been coming in from the audience today. Certainly want to leave time at the end, Chad, for you to sort of hit the highlights to just reinforce as well. And I think many of these questions maybe have already been touched upon, but it never hurts just to come back and reinforce so that people are clear about the trial. One of the first questions we have was regarding non-MELAS patient participation. And Marnie, I think you spoke to this already. It's really more about the symptomology and confirmation that you have a mitochondrial disease than it is to say you must have MELAS. That's a really important message for our audience to understand.
Yeah, I think that's a caveat we get a lot of questions about, and the disconnect between a genetic mutation and genetic confirmation and the clinical phenotype is a constant battle. When you say a patient has MELAS, what does that actually mean? Does it mean that they have a mutation, or does it actually mean that they've
Right.
had a stroke-like episode? And so what we were trying to do with the study was, again, find a homogeneous population, but also one that makes sense based on what we're trying to treat. And so we're really more focused on the clinical presentation. And so that clinical diagnosis of MELAS and having those CNS symptoms was really the focus. But we do obviously want to make sure that a patient does have mitochondrial disease and there's genetic confirmation of that.
Yeah, and Marty, I think that just reinforces something we've spent a lot of time talking about, and that's the importance of a genetic diagnosis to participate in clinical trials. And UMDF talked at the recent symposium about a no-cost genetic testing program we hope to have open very soon. But in general, diagnosis is really the start of the journey towards development of treatments and cures. Maybe you could just comment on that briefly, Marty.
Oh, I'm your biggest fan, Phil. I fully agree. I think just like the aging study he mentioned, right? Some of those people could have had mitodysfunction, just as part of aging, but genetics isn't really a part of just aging analysis right now. Maybe it should be. Maybe once we have a therapy, it will be. So I think we want to know if this therapy works for mito disease, and so this is our opportunity to learn that. It's not really just for 3243. It's not really just for mtDNA. It's for people with neurologic problems, and it really comes again to the heart of the issue of what is a mito disease? Is a mito disease an acronym? Is a mito disease a clinical symptomatology? I think we've all come to the conclusion that there's an awful lot of genetic reasons why the mitochondria function becomes impaired. And we want to make sure that the people in the study have one of those 300 plus genes have a problem. And that's what's causing the mitochondrial energy failure, and that they have some sort of neurologic problem. If they don't have any neurologic problems, this isn't the study for them. Right. But we certainly we follow many patients. So to all around the country, you have thousands that the UMDF interacts with. Neurologic problems are serious, and historically there's not one clear path forward. So we talk a lot about getting therapies in our arsenal, right? We need therapies for the brain problems in mito disease.
We really do.
We'll start to learn.
Huge unmet medical need, without a doubt. So there's another really good question that came in about where can I get evaluated, right? So obviously you don't have to go to one of the sites in order to be evaluated as potentially having MELAS or mitochondrial disease. That really falls to your primary care doctor or the mito specialist that you see. But maybe just reinforce, and Marty, perhaps just a quick mention of either NAMDAC or Mitochondrial Care Network, right, as two resources that our patients can turn to.
That's a great point. So mitochondrial disease diagnosis used to be incredibly limited in terms of who can do it. And the genetic testing revolution, the availability of mitochondrial DNA genome sequencing, of exome sequencing, of the whole genome sequencing now has really kind of liberalized the diagnostic process. There's a lot of people that can help depending on your symptoms. Here, if it's neurologic symptoms, neurologists have gotten very good at this. If you go to the mitochondrial medicine website, there's a list of all the doctors around the world who have claimed that they would like to see mito patients for whatever specialty and whatever reason. So I definitely encourage that. All of the sites involved in the trial are also clinical sites. So any one of them would agree to also evaluate you and do a thorough, careful assessment. And then on top of it, NAMDAC is the North American Mitochondrial Disease Consortium. I get confused on the total number. I think it's 17. It might be 18.
I think it's 16, but
16.
That's all right.
There's a lot of sites. So between the Mito Medicine Society and NAMDAC, there's also the Mito Care Network, which you can speak to the most, which is it's really some of overlapping sites, some others, and there's over 20, programs providing clinical care. NAMDAC is more research. So there's a lot of ways to participate. I don't think you need to even be from America. Chad can confirm. I think you have to come to America. Right. You have to physically come for the visits that you have to, but you could even participate in the UMDF or any of these centers from wherever you are. So this isn't limited. Is that right?
That's correct. Yeah. I mean, we're looking for patients from all over. Obviously, if you're able to travel and come to the clinical site, we'll work with you and facilitate reimbursement for that travel and make it as easy as possible. So
No, that's really great. So, thanks, Marnie, Chad, for sharing all that. Plus, please, audience, know UMDF always stands ready to help. If you need help finding a care center near to you, a doctor, please use our support services for that purpose as well. Chad, maybe a couple of questions that came in here I think are best suited for you. One of them being, is there any range? I know we say 18 or older, but is there sort of a sweet spot you're looking for in terms of age range, or should no one even be concerned about this? If you're 18 or older, that's meeting the inclusion criteria.
Yeah, currently it's just 18 and older. We've cast the net a little bit wider. I think there'll be some limitations in terms of how severe the disease is and whether these patients are capable of going through the assessments that are required per protocol. So, we are targeting patients with cognitive impairment, assuming that that's just kind of already diagnosed and part of who these patients are on a daily basis. But I think if they're unable to perform those questionnaires, they're unable to go through some of these procedures, they would be excluded. And I think just by nature of their ability to complete the protocol, we'll be excluding some of the maybe potentially older and more severe patients. But right now we only have a cutoff for the minimum age.
And any reason why EEG markers were excluded after what looked like promising preliminary data?
That's a great question. Yeah, so we talked a lot about EEG externally. We combed the literature in mitochondrial disease. I think the EEG that the signature that we were able to obtain in our most recent study is a fairly homogeneous, healthy aging type population. In a mitochondrial disease patient, specifically patients with MELAS, there's a high likelihood that signature may be more variable just based on the presence of previous stroke-like episodes or brain lesions, and just summarizing kind of the electrodes and brain activity is a little bit more difficult. And so we got some general feedback that in a small study such as this, that would likely be a major challenge. But it's something
maybe something you return to in a future trial or something.
Yeah,
right?
it's something I think we'll continue to look at. We have a huge EEG kind of deep dive that we're doing to fully understand it and understand how to move forward with that endpoint in mind. So
that's perfect. So Marnie, maybe back to you. A couple of I'd say operational questions, right, where the patients are trying to understand, if they're enrolled and they take part in it, particularly around the lumbar puncture. Chad mentioned now optional, but if they were to move forward to it, roughly what's the time commitment involved in actually completing that?
So the lumbar puncture itself, so that's near the end of the study. It's really just one time, and it's typically done very quickly. You would go to the radiology suite, and they would, you would be awake. There's not really any general anesthesia. They would probably use a little bit of local, and they would have an ultrasound guide and put a very fine bore needle into your canal, the same way they would really go for a woman needing an epidural. Very, very careful. They would go in, they would take a small sample and come out. So we designed it in this sort of most straightforward way. It's not going to be a medical student, meeting you, in the clinic room. It's going to be done by like the expert radiologist, in the suite, where really everything should be very, very controlled. So I think that part of it is, it sounds worse, I think, than it is. And of course, people have the right. I think, it's always a trade-off, right? When you're in a clinical study, how much data are you gathering, and how much risk are you taking? I think it's definitely more than minimal risk. Most people don't have spinal taps most days, but in a proper center, it should be very low risk.
Yeah. Any post-procedure risks that are typical for something like that? Headaches or anything that you would?
Yeah, I think so, right? Like if it's certainly always a risk to have a headache. It's also hard in a condition like MELAS where people have headaches. So we certainly understand that. Certainly, infection, if, from the site, although that would be very small. I think those are the two big ones. Chad, am I missing anything else from risk?
No, you're right. Headaches and a very minimal chance of an infection. But it's a fairly standardized procedure, and as Marnie said, it's very well thought out. But there's a risk with any of these in clinical trials, so we don't want to diminish that. But in general, it's a fairly safe procedure.
Yeah, so here's a really good question, Chad, that came in from our participants there today. If you are ultimately a part of this trial and it's successful and it moves on to another study, are you automatically a candidate or enrolled, or sort of any opportunities, I guess, that automatically come about by participating in this phase 2A?
Yeah, I mean, I think we're always looking for patients for the study in if we were to move forward in subsequent studies. So automatic is a strong word, but absolutely, I think we would want to re-enroll. There's obviously a period of time between the studies, and it'll probably work itself out just based on the time from the end of this study to the start of the next. But provided that they haven't been exposed to our drug for a period of time and they're considered a fresh patient at that time, even though they've participated in a study in the past, they would absolutely be eligible for the trial. So we would encourage any patient that enrolls in this current study and is still interested to contact sites and get involved in the next trial. We would absolutely welcome their participation.
But it is a really important point, right? There's no way to make future promises, right? When it comes to clinical research, and that it's one stage at a time. And for now, this is really about trying to find that signal, make sure it's safe, see how the drug is metabolized. An efficacy and well-structured safety trial, right, would certainly come after this, and there would be new criteria and screening involved, right, to join that.
Exactly.
Yeah, and Phil, if I could just comment, it's always hard when you design a trial, right, to know exactly where the medicine's going to work. But this trial is really like kind of going for the gold, like looking at the hardest part, the brain. But this drug should also get everywhere. So families are like, oh, or patients, we feel fabulous. I can run a marathon now. I don't think anybody will ignore what they're telling us. So the patients, opinions and what they experience will probably also guide future development. Chad, do you agree? Yeah.
Absolutely. Yeah. I mean, we're going to, like you said, this is our first study in MELAS. We have some hypotheses and we know where we want to focus, but we're not discounting any other potential. It's bioavailable in the periphery and, gets in the blood and other tissues potentially. So we're open to what the drug does, although we do have some things we're focusing on.
Yeah, and I would also just say, Phil, that it even confuses me because we have such a long history. When he says MELAS, right, we immediately jump to the genetics of it. But MELAS, they again and again, if you spell out the acronym, it's mitochondrial encephalomyopathy and lactic acidosis. So in order to get into this study, you have to have something going on in your brain and high lactate, or a history of high lactate, and so even that level has come down, I think, in the new protocol for the requirements. So again, I think it's if you have neurologic symptoms and a mito disease, they're going to see if they can help the neurologic symptoms, which is again, a great effort. Let's hope it works. Let's get there. And I think the spinal tap, just to go back, is to make sure we're giving enough. Did we give enough of a dose to give it a fighting chance?
Right. I'd like to come back to that, but let's just pause for a second. I know Beth wants to share our last poll of the webinar, and then maybe while we're collecting data, we can talk just a few minutes more.
Sure, absolutely. We actually have two polls. So one, again, is for just the patients themselves, or if you're a caregiver entering on behalf. This next poll has two questions. And then once that's gone and conversation continues, there will be one last question about the usefulness of these webinars.
Great. Looks like those have launched. So we encourage our participants to join the poll, share their answers. Marnie mentioning about changes in protocol, right? This often happens. Again, it's research, and you adjust the research based on what you're learning, right, over time. Because some of the inclusion criteria have been changed or relaxed, appropriate to say, if you've already been screened, maybe it's time to recontact one of the sites to see if perhaps now you are indeed a candidate.
Absolutely. Chad, do you want to comment on that?
Yeah, absolutely. So, we are learning as we go. This is a focused study trying to enroll a homogeneous population, but we can only get so far if it's not a feasible population to enroll. So as you said, there's aspects of the protocol that we've updated to hopefully make it more feasible and include more patients that still hopefully answer the scientific question that we're posing. But lactate levels is one, so we're still requiring elevated lactate levels during screening, but not necessarily at the level that we had initially started the study with. As you said, lumbar punctures are becoming an opt-out procedure. So if a patient was initially screened and learned about the study and determined that they don't want to participate for that reason, they can absolutely come back and be reconsidered. There have also been some changes to the protocol. This drug there's still more that we're learning about it and its drug-drug interaction potential. And so we had a long list of medications that were initially excluded that patients could not take because we didn't have data to support it. But now that we've generated data, we can remove that long list of medications, and that list is much smaller. So patients that were screened out initially because they were on some of those medications should absolutely reach out to the site again and be reconsidered.
Fantastic. Well, we are at top of our Chad, I don't know if you have a few comments that you'd like to share just in wrap. I think we've covered a lot of it. It's been a great Q&A session. I want to say from my side, thank you to the patients that have joined us today. I've mentioned this on multiple webinars. This is equal parts education as much as it is information sharing about a specific trial, learning about how clinical trials work, how they work with sites, how you can participate. It's something that our community is still very much in the early days. And when you participate in these webinars, hopefully you come away having learned something. And of course, we hope also that the information has been relevant to you and that you might be able to participate. I'll just reinforce once more, clinical research is imperative if we're going to have treatments and cures for mitochondrial disease. So absent an engaged patient community, there won't be therapeutics for mitochondrial disease. And I know that's a shared goal for everyone on this call, everyone in our community. So please think about it. And it's as much about those in the future as it is about yourself. It's a way to help bring about improvements in care for all the patients in our community now, but also in the future. So please consider that. Chad, I'm going to leave it to you to wrap with your comments. And then Beth, I don't know if you wanted me to share the poll results. Maybe I surprised Beth.
No, I had shared them. I can certainly go ahead and share the results of the last one of the usefulness, but I actually want to share the clinical trial interest one here. I'll share that right now.
Yeah.
And see that?
Yeah. So
that's what we all like to see.
This is great. Yeah. So I mean, my wrap up was, we're here for patients, and obviously we all have the same vested interest in getting patients in to answer a scientific question and hopefully move this drug forward. And so we're trying to make this as easy as possible for patients and families, and we're interested to hear feedback. And if there's questions or interest in the trial, you can go to clinicaltrials.gov, and all of the sites are listed there with contact information and numbers, so you can call the site. And we're also working on some other initiatives to kind of extend our network, and so folks are aware of the trial. But again, we're super excited to be in this space, and I want to thank Phil and Beth and Kara and UMDF for allowing us to have this platform to talk. And then obviously, Marnie, you've played a key role in this, and we appreciate your support and your additional comments on this. But again, thank you, everybody, and hopefully this was beneficial to those of you that dialed in.
Yeah, thanks very much, Chad. I'm certain it was. So again, thanks to all who attended today. This will be recorded, so if you know there's someone who perhaps couldn't be here live, this will be posted and shared. And I'm sure Margaret and Carol will be sharing that with the community where they can find the video. Let's hope that you've learned something today, and we look forward to sharing future updates with you on clinical trials. Thanks, everyone.
Thank you so much.